Hi guys, in this episode we are going to investigate the origins of behaviour in the context of its underlying genes. Now, investigating the genetic underpinnings of behaviour, as you would imagine, is quite technical and quite complex. Vertebrate genomes are inherently complex. So how do we identify the array of genes that might be associated with a particular behaviour? There's various ways you can do this, and later on in this episode we will actually illustrate one way of doing this using the study system of amphibious blennies here on the island of Guam. The reason why we might be interested in the underlying genetics of behaviour is because we would like to understand its evolution. Now, evolution operates on an animal's phenotype, its overall appearance and behaviour, but underlying the programming of that appearance and behaviour are genetics, or genes, I should say. Now, certain individuals in a population will inherently differ in appearance and behaviour, and some of those individuals will be better able at surviving or reproducing than other individuals in that population. Those individuals have what we term a selective advantage, so they're better adapted to that particular situation there and then. And those genes that are associated with better reproduction will ultimately proliferate through the population in future generations. And as a consequence, that population will progressively change in its appearance or behaviour. And this, in a nutshell, is what natural selection is and how evolution operates. So a lot of the genetic studies of animal behaviour typically focus on invertebrates, things like fruit flies or Drosophila, and in lab settings. And there is a reason for this, because obviously invertebrates and fruit flies have a very highly studied genome, and that genome is relatively simple. It's still extremely complex, but relatively simple compared to, say, a large vertebrate. So a lot of what we understand about animal behaviour comes from fruit flies. But what we often want to understand is the underlying genetics associated with behaviour in other species, in particular vertebrates, and in particular under natural conditions, how they actually are in the field. Now, this is quite difficult to do because you're removing that genetic study and putting it into a field setting rather than a lab setting. Now, in addition to most of our information coming from studies on invertebrates and lab settings, like the fruit fly, most of our information on the genetics associated with behaviour is actually the genetics of morphology. There's been a lot of work on animal coloration. Now, animal coloration is important for the behaviour in animals, particularly prey, for trying to be cryptic and avoiding predation. So it's a big deal. So studying animal coloration can be really important in that context alone. But of course, as you probably now know in the course, animal coloration is really, really important for mate choice as well. So males are typically brightly coloured and females are selecting among those males based on their coloration, its saturation and its complexity perhaps. So we have a number of studies that have investigated the genetics associated with animal coloration. Several come to mind. In particular, they've identified the specific genes that might be associated with body coloration. Beach mice, some occurring out on the sand dunes, others occurring in the grass away from the foreshore. They differ in coloration. Some are more dusty coloured, so they match the sand out on the beach. Others are more brown and cryptic coloured because they match the colour in the shrubland. Now, this is actually selection. It's the same population, they're actually the same species, and the reason why they differ is through predation. It's better to be dusty beige colour out on the beach because you're not going to stand out as much, and obviously you don't want to be that kind of bright, dusty, pale colour if you're in the shrubland because you will stick out. So those individuals will tend to concentrate on the beach, whereas the brown ones, which are particularly cryptic in that shrubland, remain in the shrubland. Now, scientists have identified that there's a particular gene or a particular set of genes that are associated with changes in coat colour. Now, remarkably, we have a different system, lizards, so entirely different animals. We find that some lizards of the same population, same species, again, occurring out on the sand. This is actually desert sand relative to other areas that are in the more shrubby areas. And again, we have brown lizards in the shrubland and dusty, pale, almost white lizards out on the desert sand. And the same genetics and the same genes associated with the coat colour in those beach mice is also underlying the coat colour of those lizards as well. So this is a remarkable example of convergent evolution. This is the independent origin of the same adaptive solution to natural selection. In this context, the adaptive solution is cryptic coloration, so either being pale and dusty colored to match the sand or brown to match the surrounding substrate in the bushland. The adaptive challenge here is predation. They're trying to not be conspicuous to visually oriented predators, in particular birds. And the underlying genetics associated with that convergent evolution is also the same, which is wonderful. So we can actually identify the underlying genetic changes that are associated with that morphology. But of course, morphology, coat coloration, it's not really behaviour, right? So when we really want to get down to the nitty-gritty of studying behaviour, we need to have a system in place that we can identify, a study system that we know well, that differs in various contexts. So not just type of habitat, but some sort of behaviour associated with that habitat. And a wonderful example of an organism that differs in habitat. So this is individuals of the same population, but in fact the same individual living in different types of habitats at different times are the amphibious fish on the island of Guam. So an individual will spend part of its time in the water, as normal fish would do, but it also spends part of its time out on the rocks in the splash zone, out in the air in what we call in a terrestrial environment. That's the same individual. So it offers a really powerful way to actually investigate the underlying genetics associated with that behaviour, because we can study the same individual in the different contexts and identify the genetic expression of that's associated with those different contexts to identify how that amphibious behaviour might be tied to that organism's genes.
So how do we actually investigate the genetic origins of behaviour? There's several different ways that you can do this. But one way that we're trying here at the University of Guam, we're actually in their marine lab facilities here in Pago Bay, is to have a look at transcriptomes. And I'll get into a little bit more detail about that in just a moment. So on Guam, as you probably know now, there are a whole bunch of intertidal blenny fishes. Most of these are aquatic, so they live exclusively in the water. However, there are a number of species that are also amphibious, and there's actually a single species here on Guam of the genus Ulticus that's exclusively terrestrial. It never goes back to water. For this trip, however, we're focused on those amphibious species. So we're particularly interested in the 3 amphibious species that have independently evolved the ability to come out of the water. And what we're doing is we're collecting a whole bunch of individuals for each of those species from Pago Bay, which is literally just 50 meters down from the marine labs here. And we're putting them into a very simple experiment. And that experiment actually just set up here behind me. We're going to run this in about half an hour's time. There's a series of tanks, and all of the fish are individually put into each of these tanks, which are full of water. So this is the simulating of the aquatic part of their lifestyle. And then after a certain amount of time, we will then transfer those individuals into new tanks Some of those tanks will again have water in there and act as controls, but other tanks will actually only be misted, so there will be no water in there. And this is to simulate the terrestrial lifestyle when they come out of the water. And then we leave those individuals in for a certain amount of time. And at the end of the experiment, we then collect some tissue samples. And then we can have a look at the transcriptomes of those different individuals. Now, to put this into context, I'll put a very basic overview of how genetics and transcriptomes and messenger RNA works. So, as you know, DNA is in the nucleus of our cells, right? So it always stays there. So we have messenger RNA coming into the nucleus, binding with the DNA, reading the code, and then moving out of the nucleus into the cell proper. And in that cell, the messenger RNA then encodes the production of proteins, lots of different types of proteins, which is essentially the underlying process of life. So what we're actually doing in this experiment is we're comparing the transcriptomes, or the differences in proteins that are actually being encoded in real time. So what we will find here is that there will be some genes that are upregulated, some genes that will be downregulated. So there'll be a high production of certain proteins and there'll be a low production of certain proteins associated with being out of the water. And when we compare the transcriptomes to those that are in the aquatic treatments, we can then identify which genes are likely to be associated with being amphibious.